Hello everyone, welcome to another video. This is going to be on translation. Previously, I discussed about DNA replication as well as transcription, so you guys are always welcome to check that out before this, but let's get into it. So what is translation in its simplest form, description? Translation is a process by which mRNA is decoded to create a protein. Essentially, previously, we have this polypeptide chain that is grown in terms of subunits called amino acids. So as segments of the mRNA are decoded, the polypeptide chain gets longer. That's because, again, with the subunits of amino acids, as more segments are decoded of the mRNA, there are more amino acids being bound to the chain. But essentially, what is the specifics of the process? And we have to consider initiation. That is the very first step. With initiation, the ribosome is formed. Initially, when I learned about translation, I thought the ribosome is just this structure that was always there. But in fact, it is composed of smaller and larger subunits. What occurs first is that the mRNA passes through and binds to the smaller subunit of the ribosome. And we also have this tRNA, the very first tRNA coming in with its anti-codon, binding to the mRNA with carrying its amino acid, which is methionine. And then, as you can see in the diagram, this big piece of the ribosome just suddenly binds. And that is known as a larger subunit. That occurs because of helper proteins, but what you need to know is that we have this smaller subunit with the mRNA binding to it, and as well as the first tRNA coming in. Then we have the larger subunit coming in and binding to the very first tRNA at the P site. That's the P site is of the ribosome, just in case you're wondering. And we'll get into that right now, actually. So now let's consider elongation. That is where the polypeptide chain is growing, and that's where new tRNA is coming in. So of course, when initiation first occurred, the first tRNA was attached or connected to the P site of the ribosome. And that's where our starting amino acid was. So when the new tRNA comes in, it comes in through the A site. So if you analyze this diagram here on my right, and hopefully you can see my cursor, the A site is where this new tRNA is coming in, as you can see. And the P site is where this original tRNA is. And with it is the polypeptide chain. So how this works is that this new tRNA, again, coming in through the A site, the P site, the tRNA at the P site will break off the polypeptide chain. This amino acid will come in with the tRNA and bind to the P site. So essentially think of it as, okay, A site, then this amino acid will bind to the P site where the polypeptide chain ideally is and will grow. And then this tRNA, since it broke off, will now move to the E site. And that is where it will exit out, will move out of the ribosome to carry another amino acid back through the A site. But that's the key. You just have to consider that new tRNA comes to the A site. The previous tRNA at the P site will break off from the polypeptide chain, move to the E site, and exit out of the ribosome. And it will continue, of course, until the polypeptide is finalized, and it will break off to then go through modification. Now, how does tRNA bind to mRNA, though? And that is really important to consider. So if we see here, and we know that mRNA is composed of codes, right? But we break this down in terms of what are called codons. So a codon is a series of 3 nitrogen base pair codes. So for example, UGG would be a codon, AAA would be a codon, AUU would be a codon, but let's, for example, consider AAA. That's the codon of the mRNA, and the tRNA, it does carry a 3 nuclear nitrogen base pair, but it is not called a codon. In fact, it is called an anti-codon. It carries the complementary base pairs of the codon from the mRNA. So for example, again, AAA, that's the codon of the mRNA. The anti-codon would be UUU, because A is complementary to U in case of RNA, which would be uracil. GAU, for example, complementary would be C, because G binds to C, A would bind to U, and U would bind to A. Now, the very last process of translation is termination. So this occurs when the ribosome shifts towards a stop codon. When we reach a stop codon of the mRNA, everything breaks off. So the smaller subunit, the larger subunit breaks off, and even the polypeptide chain breaks off because it needs to go through a modification process, correct? But that occurs, again, keep in mind that occurs when we reach a stop codon of the mRNA. And lastly, I wanted to end off with the codon chart. So again, a codon is a 3 nucleotide base pair, and the anticodon would be the complementary of that, right? And that is, or the tRNA holds that. But if we want to analyze the codon chart, we have to consider, and this is very important, we have to consider the mRNA code, not the tRNA. For example, the mRNA code reads AUG. Okay, we read that. So AUG, we have A here, we have U here, and then G, it's methionine, which is our start codon. But the complementary of that would be UAC. That would be the anticodon. And that would work, actually, frankly. U here, A there, and then U here, A here, and then C, that would be tyrosine. But do not read the anticodon. Very important, and I made that mistake myself. You have to read the mRNA code. Whatever that is, even if it's CCC, for example, you read that. So keep that in mind. But before we end the video, just a quick review. Very first thing that is important is that translation produces a polypeptide, which would become a protein, a finalized protein. The 3 stages of translation are initiation, elongation, as well as termination. When reading the codon chart, you must read the mRNA code. Always keep that in mind. And the ribosome will shift and read the mRNA. It's not the mRNA that moves technically. It's rather the ribosome which makes the mRNA kind of have the impression of moving forward. But the ribosome reads the mRNA code, and until it reaches a stop codon, that's where everything breaks apart. The polypeptide chain becomes free. And that, everyone, is the end of the video. Thank you so much for watching. I really appreciate it. Please hit that like and subscribe button, as it helps always and motivates me to continue making these videos. But for now, I'll be taking a break from biology and moving on to calculus and then physics. But until then, thank you so much for watching.